Assalamualaikum dan salam sejahtera kepada semua yang mengikuti siaran webinar bersiri Jabatan Pengurusan Tanah pada pagi ini. Bagi makluman semua, siri webinar ketujuh ini merupakan webinar anjuran Jabatan Pengurusan Tanah dengan usaha sama Unit Jaringan Industri dan Masyarakat, Fakulti Pertanian UPM. Jadi bersama dengan kita pada pagi ini adalah Profesor Madya Dr. Tan Geok Hun dari Jabatan Pengurusan Tanah Fakulti Pertanian UPM. Jadi sebelum beliau menyampaikan webinar pada pagi ini, ingin saya mulakan dengan sedikit pengenalan latar belakang ringkas. Kita mulakan, iaitu Profesor Madya Dr. Tan Geok Hun dilahirkan di Rantau Panjang, Kelantan. Beliau menerima pendidikan awal di Sekolah Jenis Kebangsaan Cina Hebun, Rantau Panjang dan menyambung pengajian di peringkat menengah di Kota Baru, Kelantan. Pada tahun 1996, beliau memulakan pengajian di Universiti Pertanian Malaysia, seterusnya menyambung pengajian ke peringkat master dan PhD. Jadi setelah tamat pengajian peringkat PhD pada tahun 2006, beliau berkhidmat sebagai asisten profesor di Universiti Tunku Abdul Rahman sebelum berkhidmat di Mardi sebagai pegawai penyelidik kanan. Beliau juga mendapat pertukaran dalam perkhidmatan pada tahun 2013 dan mula berkhidmat di Fakulti Pertanian UPM sehingga kini. Penyelidikan utama beliau ialah dalam kajian terapeutik bacteriophage terhadap pelbagai jenis penyakit dalam tumbuhan dan haiwan, pemeliharaan dan pemeliharaan mikrob serta penggunaan mikrob berfaedah dalam pertanian. Beliau juga telah menerbitkan lebih kurang 90 penulisan artikel jurnal dan kertas persidangan. Beliau adalah ahli kepada Asian Consortium for the Conservation and Sustainable Use of Microbial Resources, ahli Asian Network Microbial Utilization, ahli dalam networking, capacity building dan Education Committee di bawah World Federation of Microbial Culture Collection serta ahli kepada Malaysia Society for Microbiology. Beliau pernah memegang jawatan sebagai penyelaras unit Microbial Culture Collection di Institut Bioscience UPM dan dilantik sebagai penyelaras untuk koleksi kultur mikrob pertanian di peringkat fakulti pada bulan Mei baru-baru ini. Jadi tanpa melengahkan masa lagi, dijemput Profesor Madya Dr. Tan Geok Hun untuk menyampaikan webinar berjudul Soil Health, Prevention is Better Than Cure. Dipersilakan Dr. Tan.
Thank you very much, Dr. Arina, for your kind introduction. First of all, good morning everyone and salam sejahtera. First of all, I would like to thank Faculty of Agriculture for giving me this opportunity to share some of my research work in this webinar. Okay. All right, before we go further, okay, so let me explain why soil health is important. Prevention of diseases is better than curing. Okay. All right, so as we know that soil is very important to all living organisms, okay, including us as a human, because we depend on soil to get some food from agriculture, okay. So when we are talking about soil health, so it means that ability of soil to offer a broad range of ecosystem, a balanced ecosystem, and also agronomics resources, and some of the activities to sustain the environment quality and health. When the soil is healthy, all right, so many of diseases can be eliminated. Prevention of disease in the soil is better than apply treatment, okay? When we can see many cases or news regarding a lot of crop being attacked by different types of plant pathogens. And you can see all those plants, when human or people detect, that means it's already at the stage 4. That means either you have to clear the whole plots or you have to use some of the treatment to cure the disease. So why not we start by looking at the soil health and then we prevent it before the disease occurs. There are a lot of plant disease originally come from the soil because soil also has a very good habitat for them to survive. These soil-borne disease usually enter the plant through the root system. All right, somehow the root have some wound and this bacteria or fungi, they will enter it and then penetrate into the xylem or phloem of the plant system, and then slowly they will destroy the whole plants. So when we see, all right, see all those plant is already dying. Disease development depends on 3 factors. The first one is soil, all right? So soil that I mentioned just now, they play very importance. If let's say the soil is healthy, then no others bacteria or disease can stay there because indigenously we have a good microbe inside there as a protector. The second factor is the virulence pathogens. All right, so all these pathogens are potentials to infect or attack the plant and then cause a lot of loss to the country. And the third factor is the susceptible of the host. If the host is healthy, all right, like us also, if we are healthy, all right, so whatever disease is very difficult to attack because we have our own immune system. It's same as a plant. Plant also have their defense system. But then if they are very susceptible, easily to be attacked by the bacteria or any pathogenic microbe, then they will receive and then attack, go inside and the whole system will destroy. So this is the 3 factor that very important. So the failure of bacterial disease management in agriculture usually caused by several factors. All right, so first will be lacking effective or bactericide against the disease. All right, so now we have a lot of problem, especially in our country because the weather, the climate is very suitable for microbe to grow happily. Okay. Second will be high pathogen variability. All right, so the soil, the condition as we have right now, they can grow happily and then will attack all those plants easily. Third will be rapid populations build up of pathogen under optimum condition. Okay. Fourth, mutation rate resulting resistant developing. And lastly is the high mutation rate result in the bacteria overcoming. Inside the soil, however, we also have many potential microbes that works antagonistic with the pathogenic bacteria or fungi. They release certain enzymes which inhibit the growth of pathogenic bacteria or fungi. In the soil, all right, in the balanced ecosystem, all this happen. That means they have a good microbe and bad microbe. They will antagonistic or they will like prevent or work. If let's say the populations of good bacteria higher than bad, then that particular area is safe. However, if we apply somehow too many of pesticide into that land of soil, so when you apply the chemical thing inside, you will alter the balanced ecosystem and then slowly the bad microbe will increase the population and then they will attack the good microbe. So this is a very stable situation or balanced situation between the good and the bad, as same as our digestion system as well. So that's why we encourage people to drink a lot of Yakult because that is a good microbe in order to protect your gut system. Okay. They also have certain viruses we call as a bacteriophages, which they only infect the bacteria. And this phenomena was applied as a potential biocontrol agent in disease management. Okay. So previously, people using copper product and certain antibiotics for disease problem. However, as we know, this copper can cause some environmental problem. They will build up a toxic level in the soil and also can trigger the resistance of certain pathogen. And in order to change, and then they may change the balancing as I mentioned just now. Okay, so the build up of those chemicals actually for long run is never bring any good impact to the environment. So what is bacteriophages? All right. So bacteriophages, or short form we call as a phage, is a one type of virus where they can infect bacteria than archaea cells. All right. So nowadays, when we're talking about virus, many people scared of it. All right. Virus is something specific and then they only attack that particular cell. For example, now we have COVID-19. All right. So this COVID-19, they only attack the respiration system. All right. That's why when we see this particular virus, they won't attack other parts of your body cell. For example, let's say the cell, the virus which attack the skin or the level or the colon or whatever, they won't attack other parts of the body. So they are very specific. So these phages as well, they are not harmful to human, animals, or plant because we isolated them from the environment and also from our gut system as well. So the potentials to be used as a biocontrol in nature. So basically, we found there are three types of phages. Those attack bacteria, we call as a bacteriophages, which attack or infect the actinomyces, we call as a actinophages, and phage which infect fungi, we call as a mycophage. All right. So I've been working with phages for more than 20 years. All right. So my PhD also doing on phages, but on other type of the phages. Basically, phage is more, how to say, they have therapeutic, that means curing the disease and also display. I will going to explain in detail later on. So let's have a look on the phages structure. All right. So I think if you watch a Hollywood movie, somehow some of the alien, they will draw something like a virus. So this is the phages. All right. So you can see they are very simple. Okay. They got the DNA inside the capsid protein and then the tail. All right. And then some neck connected between the head and the tail. Right. The outer shells of the phage is composed of various protein. With the capsid head, the function is to protect the viral genome inside there. Some phage are minimal genome, only 4 protein, and then they can form a capsid head. And then the others can be hundreds of genes. So this depends on the species and subspecies of the phages. So previously, in medical especially, they use the antibiotics to cure some of the bacterial disease. For example, like MRSA. However, recently, they got a lot of resistance from the bacteria. That's why we are slowly to use phages to replace the antibiotics. Let's say if you got sore throat, you see doctor will give you some antibiotics and ask you to finish, let's say, within 3 days or 5 days. All right. So how the antibiotic difference from phages? So antibiotic, actually, they have a broad mechanism. They kill all kind of bacteria, including the beneficial microbe as well. Okay, they kill everything, they don't care. But then for phages, they are narrow mechanism, only eliminate the specific host. Why I said they only eliminate the specific host? Because they have a specific receptor with the particular bacteria cell. Okay. All right. So this is a little bit the history of phage technology. All right. So they got two different sections for phage technology. Phage therapy, which we use as a biocontrol in human, in animals, or in plant. And another one is the phage display. All right. So phage accidentally, all right, been discovered back 1896. All right. So they observed some antibacteria against Vibrio chlorella. So everyone know what is Vibrio chlorella? This is the bacteria which cause diarrhea, or Malay we call as a taon. All right. So when they infect you, you will have a very bad diarrhea. All right. And then it was discovered first back to 1915. All right. And then 2 years later, okay, and then used as a therapeutic agent by Felix Harriel. For agriculture, in agriculture, phage was proposed as a plant biocontrol by Moore back 1926. That means it's almost 100 years that phages has been used as a biocontrol agent. But why we did not use this as a commercial? Then I will explain at the back of my presentation. Okay, so just a little bit on what is phage display. All right, so this technique can be used to study the receptor antibody binding site. All right, so to determine the epitope mapping, this is more to antibiotics and also antibody work. And also to improve or modify the affinity of protein for their binding partner, vaccine development, and also drug discovery. So recently for COVID-19, all right, US was granted a big money by using phages to display some of the surface antigen, or we call as a spike of COVID-19. Okay, so that is the new development. So that means the phages will carry the antigens of the COVID-19, and then this can be used as a antibody detections or any vaccine development. For diagnostic reagent for vaccine and detection, identify the peptide ligand with random peptide library. Okay, and also ligand interaction. Okay, just show some of the work being carried out previously. Okay, so I'm working on hepatitis B virus previously on surface antigen, core antigen, and also E antigen. Okay, so what we did is we display these three antigen on the surface of the M13 and also T-type phages. Okay, so this one we use as a detector for the antigen and also antibody. Okay, so when we talking about virus infection, sometimes we did not detect the whole particle of virus. Okay, we only detect certain protein which release in our blood system. Okay, so all this is we call all these protein, when they release, we call as a antigen. Okay. All right, so come to the phage therapy for agriculture. So they have applied these phages for many bacteria as well, pathogenic bacteria. For example, in the Pseudomonas syringae in leeks, Okay, and then Pseudomonas thalassei in the mushroom. So you can see a dot here. All right, it's a attack, in fact, by this Pseudomonas thalassei, and then in the apple and pear by Irvinia chrysanthemi. So this is quite famous in pineapple as well, right? So they attack and then they will cause some smelly and rot at the middles of the pineapple plant. Okay, and then Sclera fastidiosa on the grapevine. All right, and Rhizonia solanacerum in tobacco, in tomato as well. Okay, and then Peptobacteria carotovum on the lettuce, and Dickia dadenti in black disease of potato. And also in our country, they attack the papaya. So papaya problem. Okay, so this is the main pathogenic bacteria which attack the common crop. However, all these phages has been used and tested potentially to control all these diseases and is already registered under company under US. Okay. So let me explain a little bit on the life cycle of bacteriophages. So bacteriophages, as we know, they attack the bacteria. They have lytic cycle or lysogenic cycle, and some are capable to carry out both. That means can have lytic cycle and also lysogenic cycle. When infections of cell by bacteriophages result in the productions of new viron mean the new particles of virus. So this we call as a productive. That means they produce the other offspring more and more. So lytic phages, immediately they kill the cell. What is the difference between lytic cycle and also lysogenic cycle? For lytic cycle, when they attach, then they directly kill the whole cell. For lysogenic, they just inject the DNA into the bacteria genome. And this DNA will replicate together with the bacteria genome. And then the bacteria genome will harmonize living as usual. Okay, so that means they did not directly kill the bacteria cell. So the lytic can quickly reproduce within and lyse the bacteria host, growing exponentially in the numbers. For example, let's say one parent of phages, okay? So they attack the average about 200 daughters per cycle. First cycle, they produce 200. All right, and then each daughter infect and kill the bacteria cell. So there will be the second cycle, 40,000 at the end of second cycle, and 8 million at the end of third cycle. And then after fourth cycle, you will have about 1.6 billion and so on. So this process will be carried out. So this is a very good way how you can prevent the soil from being infected by the pathogenic bacteria. Let's say you have a good phages and you apply to the soil before planting. All right, so you already have all these populations standby. All right, so once this pathogenic bacteria come and attack, then they cannot penetrate the surrounding of the plant. Okay, for example, let's say you are doing on the Xanthomonas oryzae on rice, for example. Before we plant any new season, it's good to apply these phages in the soil. So as a protector or as a guard to take care before this bacteria can attack. Okay, so this is how prevention is good than curing. However, it depends. The numbers, this one also is depends on the populations and also the species and also the condition. All right. So this is how the mechanism of infection. All right. So first, I said the phages is very specific because you can see this is the tail. They have a very specific receptor with the bacteria. Okay. So how they work? Firstly, they will go into find the suitable receptor at the bacteria surface. Okay, and then after it's suitable, it's something like the lock, the key together is matching, then they will try to inject the DNA into the cytoplasm of the bacteria. Then after that, this will be, once the DNA inject into the bacteria, they will take over the machinery. Supposingly bacteria, they will duplicate and then produce their own genome. However, when we take over by these phages, they will produce the phage DNA rather than bacteria DNA. Then after that, each of the capsid protein will be produced, and then they packaging. They call it as packaging process. That means the DNA go into the capsid and then package become a new viron. Then after that, they release the endolysin. Is a one type of enzyme to break down the cell wall or membrane cell of the bacteria and then release into the environment. And each of the phages will go and attack the other same species of bacteria again. Okay, then so this process will repeat over and over until the whole pathogenic bacteria clear, nothing, then this virus will stay calmly, become dormant. Okay, so this is the process how it works. So this is the real environment in the soil. All right, so you can see before treatment and after treatment. So this is the pathogenic bacteria. Okay, so as we know, bacteria also, they can multiply very fast. Within 1 hour, you will have a millions of it. Okay, so when this without the phages, so they will multiply, attack the plant, and then slowly will attack to the root, the leaf, and so on. So when you apply the treatments or prevention, so this bacteria will be attacked by the phages. So slowly they will reduce the populations of the pathogenic bacteria. So this is how we apply as a biocontrol prevention. Okay. Okay, let me share some of the research which have been carried out from the previous time. All right, so the applications of phages on bacteria leaf blight, BLB in rice. Okay, so as we know, bacteria leaf blight or penyakit hawa padi, all right, is a gram-negative bacteria caused by Xanthomonas oryzae, Pata wap oryzae. So this is the most serious disease in rice, Southeast Asia. Okay, they can infect rice at any stage of growth. They penetrate into the plant through the wound of the roots or leaf. Okay, so this is very quite dangerous. And also when the plot attacks, so you can see, okay, this one, sorry, you can see the plot. So this is the epidemiology cycles of the bacteria leaf. So you can see when attacked, you'll see the yellow color dry at both sides of the leaf. Okay, so this is the how they attack the plant. All right, so this photo you can see, let's say you go to the field, rice field, when you can see a spot yellowish there, most likely there is the Xanthomonas oryzae. So you can see here. Okay, so from this study, the way from this study we have to isolate the Xanthomonas. So I would like to share some experience by isolating the Xanthomonas. Normally, pathogenic bacteria, they prefer to grow naturally in the plant cell. Okay, for example, let's say we go and search all those microbes, okay, and then they can easily grow. But when we bring it back to the lab, all right, and then grow in a certain agar plate, so they are very difficult to grow on the plate. That means because on the plate and the natural ecosystem, natural plant is slightly different. So it's very hard to maintain all these pathogen. So what we can do for those doing on Xanthomonas, you can grow your Xanthomonas in the rice plant. That means maintain it in the rice plant. First, because you can maintain the virulency of the pathogen. If let's say for long run, this pathogen, if we transfer to the plate for long run, the pathogenicity will drop. Okay, so this is the Xanthomonas, red yellow color on the plate, and is a gram negative. So this, from this study, two potential phages has been isolated. Actually, we isolate a lot, but then screening, I think a lot, that means we have about 100 something of the phages, but then only 2 of them work effectively. And then we look at the TEM, and then we can see this is a big head and small tail over here. Okay, so this A and B plate showing the plaque forming by the phages. So normally how we do is the plaque clear zone represent the particles of phages. Okay. All right, so from these two also we managed to identify, and then the sequence, all right, the sequence of the phages. So we identified using next generation sequencing. Okay, so N1, they have the genome sequence about, for N1 phages has the genome sequence about 41,152 base pair, and N3, okay, is about 33,454 base pair. So these two is a new phages which been recovered. Okay, discovered. So all the genome sequence was deposited under NCBI. All right. So this work we were going to pattern all this under the UPM. Okay, so second study on the bacterial wilt disease in tomato. All right, so I also working on Ralstonia solanacearum. This also the most devastating tomato disease in lowland Malaysia. Normally, lowland tomato has a problem, okay, because of the weather and so on. And then this is Ralstonia solanacearum is also a soil-borne gram-negative bacteria. So the infection takes place through the root as well, and then have a very strong tissue-specific tropism within the host. So they invade into the xylem system and wilting. That means cause the wilting of the plant. All right, so it easily spread through the contaminated soil and irrigation water. And these bacteria, they can survive for many years with alternate hosts. Alternate hosts, that means they can attack the other plant as well, which under Solanacearum species. Okay. So this is the application. So this is the another types of bacteria. Okay, so what we can see first, all right, so when we bring the sample back to the lab, the first thing you want to see either this plant being attacked or infected by the bacterial wilt disease. The first thing easily you can observe, you cut the stem, all right, and then put into the distilled water. After a few seconds, you can see some white string moving out from the stem. So this showing that this is bacterial wilt disease. So you can see we call as a ooze, OOZ, coming out. Okay, then when we streak, okay, when we streak on the TTC medium tetrasodium, you can see 24 hours later, you can see some pink color appear on the plate. All right, so be careful these two color. That means if they are very virulent, that means very strong to attack the plant, when the colony form is only a pink dot in the middle of the white edge. This means that this bacteria is very virulent. If after a while, you leave it there for a few plate subculturing, so you can see some of the red color. So if you found that the red color, very red color without any edge of the white color beside, then that means the virulency or pathogenicity already reduced. So this is how you observe your pathogenic bacteria, either they still virulent or not virulent. For those who are working with all these pathogen microbe, because the importance, we want to find the biocontrol to control this bacteria. If the bacteria, the pathogenicity already dropped, so it's no point that you use the biocontrol to control them. Definitely you will get a very good result. So what we need to do is we should find those very high pathogenicity. Okay, so this is some of the experience that I would like to share. All right, so we found some of the phages as well. All right, isolated, and then all this is under TEM, and then these phages was applied on the tomato plant. All right, so from this graph, we can see some comparison. Okay, so T1, this time we incorporate the fertilizer with the phages. So from the treatment T1, all right, so the T1 is from the study we managed to get, and then this is how the disease scoring, all right, in T3 is higher because inside T3 there is no phages inside. So for T1, we incorporate with the phages. All right. So for the third application is on the stem rot disease in dragon fruits. Okay. So dragon fruit, there are three major fruit. I think everyone know dragon fruit. So normally these dragon fruit, they are very tolerant to the pest. However, there are certain pests that can associated with this, okay, due to the unfavorable climate and management of the crop. So again, the genus Xanthomonas, but this is not Xanthomonas oryzae, this is a Xanthomonas capestrae. Okay, so this is the different types. Same genus, different species. So this infection start from the injured area. Okay, so especially when the stem tissue by the insect or any anthracnose attack. So after that, they will have the secondary infection by this Xanthomonas capestrae. So yellowing of tissue followed by softening and smelly rotting of tissue is the symptom of rotting. At one attack, total rotting of fleshy, succulent parts of the stem leaving the main vein. That means if attack the highly, you only can see the vein of the dragon fruits. So normally, this problem will be controlled by pruning of infected parts and then apply with copper. However, after a few months later on, this Xanthomonas capestrae will come back. So what we did, we apply, all right, the phages cocktail with the fertilizer. We apply before, okay, and then some which already been infected, we spray at the infected area. Okay, so after a few 2 months later on, so you can see some of the dryness of the trunk of the stem here. Okay, so this is beginning of the study. So this is the treatment without, and this after a while we apply with the phages, so they become dry, and then after that the new shoot will come out. So this is the application, because the area that we have studied is already full of infected with Xanthomonas capestrae. So what we can do, just to apply as a treatment in order to save the other parts of the tree. Okay. All right. So the second mechanism of infection is by lysogeny. All right. So just now we already have a look how the lytic phages work. So the second phage, this one also involve the same thing. The phages also attach to the bacteria surface. All right. At the same time, they also inject the DNA into the bacteria. However, all right, the phage DNA incorporated. So you can see this the green color. All right, green color is from the phages. So when the bacteria multiply, at the same time, they will multiply the phages DNA as well. And the protein also expressed together with the bacteria protein. Okay, and then they won't produce any offspring or any new viron inside here. So they will live harmoniously inside here. So the DNA will be copied from one bacteria to another and so on. So this will be continuously. Okay, so from this study, okay, during the integrated portion of the lysogenic reproductive cycle, the viral genes are often expressed. All right, that means the bacteria, when they express their own protein, they also express the phage protein at the same time. So RNA polymerase of the host cell read the viral gene just as they were host gene because they thought it's the same because it's already incorporated into the same genome. So this caused some changes of the host genome and expressions of protein which have potential to exhibit certain growth in antagonistic SADI or Ganoderma body nets. So actually, this is accidentally found out. Okay, so I working on this bacteria, and this bacteria I just have a try on the Ganoderma. And after 5 days, then I monitor until 20 days. Then I can see some changes of the Ganoderma on the plate. Okay, so this is the inhibition zone by this bacteria, which that means some changes on the expression protein over here. So this is the control. So on the top side also, and this is the bottom, so where I can see some burning area. So I do not know what happened. All right, so this is the some accidentally found out. Okay, however, we still need to do a lot of work because this one we need to know what actually been expressed of the bacteria during the process and how the mechanism involved until the Ganoderma did not grow beyond the size. Okay, so this is something interesting. All right, so for the product development, all right, phage can be applied for potential biocontrol in the plants. The application can be applied on solid fertilizer or liquid. Applications of phage is more prevention than treatment. So mean application onto the soil than to be used as a safeguard for the plant. So basically we are target as a prevention rather than treatment. Okay, just to share, the project has been carried out with collaboration with Cosmos Industry and Mardi. So this is under Techno Fund. We have 1.5 million. So this is the product, have 4 combination called as a smart fertilizer or a chemical NPK, organic, slow release, improve soil structure, and have some effective microbe and also phages add-on as a prevention of disease. So this project was funded by MOSTI back 2014. Okay. As a challenging for the phage therapy, as we know, phage has been used since 1926. All right. However, why is it not used widely? Okay, so the first will be the resistance of the phages. As we know, virus, they can mutate very fast. Okay, same as our COVID-19. How many substrain already been changes? So this is the resistance of the phages. The second is the criteria for selecting suitable phages. For the Xanthomonas work, we have 100 plus almost 200 of phages, but only 2 potential to be used as a biocontrol on Xanthomonas. Okay, and strain variation. All right, certain phages, they only targeted on the genus species. Okay, and then they are multi-host range. So that is also some of the potential, because when we going to apply to the natural environment, we must think, if I use only one phage, if that particular phage mutate, what will happen? So it's good to have a cocktail of the phages plus some of the wild type, which have potential to control that particular pathogens. Okay. Persistence of phage in the xylosphere and also rhizosphere. Okay, for example, certain applications of the phages on the leaf, for example, or the root system. Because as we know, inside the soil, we got so many microbes, okay, and they are very strong enough. So when we want to apply some new something new into the soil, all right, the indigenous will try to protect it. So the populations that we're going to add in must reach certain level. Let's say 2 liter, it doesn't work. It's too much, you're going to waste it also. All right, so we have to persistently and make sure that they always there with the certain amounts of PFU. Okay, so that's why. So recommended is about 10 power of 6 or above is suitable for prevention diseases. Okay, and then resistance to the phage. So bacteria also will resistant to the phages. They will try to escape. Okay, try to escape. For example, let's say I give example bacteria, bacillus, for example. No, maybe Pseudomonas. So when Pseudomonas, we have some phages can attack these Pseudomonas, and then these Pseudomonas, they will try to mutate as well, so that in order for them to can escape from these phages. So when they try to alter, mutate, and these phages may be not suitable for these Pseudomonas anymore. So this is some of the challenges that we need to consider. That's why it's a lot of work to be done before we come out a suitable product to apply as a prevention. Okay. All right, I think that's all. So any question? Thank you for your attentions. Okay, thank you very much.
Okay, thank you, Dr. Tan, for the sharing session. So now we can begin with a question and answer session. Any question from the viewer? Feel free to drop questions in the comment section. It looks like the viewer has been enjoying watching and listening to your webinar, Dr. Tan. Any questions so far? No? Okay, so my quick question would be, is the product will be readily available in the market?
Yes,
can be
because
obtained readily available in the market. Yep.
Because this collaboration between All Cosmos and Mardi and also UPM. So basically, it's available. So the ownership, that means the production side is from the All Cosmos Industrial. Yes.
Okay. So it looks like we don't have any additional questions from the viewers. Okay. So once again, I would like to thank Dr. Tan for her sharing session today. Okay. And we will have another webinar session after this in another 2 weeks time. Okay. So till we meet again next time, and thank you everyone, and stay safe.
Thank you very much, Dr. Arina and audience. Have a nice day. Bye-bye.
Bye-bye.
Bye-bye.